So our next section for the animal kingdom is invertebrates with a true coelom. So coelomic animals are divided into two groups based on embryological evidence. And is that right? Just a second. Yeah, we talked about that last. Okay. I had to double check. I thought I had skipped something, maybe. Okay. Sorry about that. Back here. Okay. So this embryological evidence uh, has to do with where the mouth forms on the blastopore. Okay. So I'm going to pull up this slide, which is if you're going through, it's 3 slides after, basically, I think three, what I just had up there. So advance a little bit. So, we've talked about this a lot, that the egg and the sperm unite to form a zygote. And that single cell, that first single cell, you've got one haploid cell with half the chromosome number, the other haploid cell with the other half the chromosome number unite. Now you have a full diploid cell. And it's going to start dividing uh, mitotically. Okay, so one cell will divide to two, and then two's gonna make four, and four will be eight, and then 16 and 32. It keeps on going. Okay, so uh, that division, that mitotic division, and doubling very rapidly once it starts. Okay, it forms this ball of cells. Okay, and at some point we get this ball of cells forms this kind of little, it's like, okay, I don't know, analogies. Take a ball of dough, play-doh, clay, whatever you can relate to there, and stick your finger into it and make an indentation. Okay, and you can see we've got two different indentations here. They have different shapes, actually. Okay, but that's what will happen. The outside of that ball kind of starts to turn in at a point into itself. And that opening created by that turning in, okay, is called the blastopore. And this pocket that's created by this turning in is the primitive gut. Okay. So what we have what's called protostomes and deuterostomes based upon what happens with this blastopore. If the blastopore if the blastopore opening becomes the mouth, then we call these protostomes. And our phyla that are protostomes are mollusks or mollusca, annelida, and arthropoda. Okay, the scientific names are mollusks, annelids, arthropods, as our more common names. Like, yeah, differences in A and an S, I know. Okay. Um, and then the anus opening will develop later. Okay. The blastopore here is going to become the anus instead, and the mouth will develop later. Okay, elsewhere. Um, and those organisms that have that happen are termed deuterostomes. And that grouping, the two phyla there, are Echinodermata and Chordata. And that's us. We are in the phylum Chordata. Okay, so and I think with that I have pretty much just summed up okay, what's on these two slides using the picture there. So I will move on uh, into talking about each of the phylums. So let's talk about mollusks first, phylum Mollusca. There are three distinct parts to all mollusks. We have a visceral mass, which is the soft portion that contains the internal organs, the foot, and that's the muscle that's used for locomotion. And we have the mantle, and that's a membranous or muscular covering that encloses viscera and may secrete a shell. Uh, we see cephalization in many of these. Uh, they have eyes, and they have different types of sense organs depending on which mollusk you're looking at. They're highly diverse, and they're adapted to many modes of life. Okay, so we have different characteristics. We have gastropods, cephalopods, and bivalves for classes. Okay, so kingdom, phylum, class. So kingdom Animalia, I'm mentioning here are phylum Mollusca, And then in that, we're actually going to talk about classes. Didn't do that with plants. So I didn't, get down into the classes. But with animals, here I am. Actually, none of the other kingdoms we've talked about have I taken you down into the class level. We've just talked about different phyla. Okay, so it's a little different, but we have Gastropoda, Cephalopoda, and Bivalvia actually is our names. So here's some pictures of examples of these groupings of this. This is the whole phylum. We have it's called a chiton. We have a chambered nautilus, a scallop. We eat those, okay, and a Spanish shawl. Um, what you have here in italics is the genus name. You don't have a full scientific name here, just genus name. That's not something I will ask on the test. This is multiple choice only, so there's no short answer bonus of writing scientific names properly or anything like that, like you saw on back on test 2 before spring break. I think I even had some on test 1, but okay, Gastropoda includes snails and conchs or conches, depending on where you are located and what colloquialism you speak is how you pronounce that. I've been told. Okay. Um, I believe that in the Caribbean islands referred to more as conchs. I think that these smaller varieties that are found in coastal U.S., southern coastal U.S., native used a lot historically by native people, referred to them and call them more like conches. So anyway, there's different pronunciations. Don't know that one is more accurate than the other. Okay, so these have ventrally flattened foot that moves by contractions. And so if you've ever seen a snail, observed a snail, watched a video of a snail, then you've seen that motion. Okay. Most people aren't familiar with conchs or conches, all right, other than maybe the shell for decoration. But uh, there's been probably some opportunity for you maybe to have seen a snail. So you know how it moves along on that little foot. Okay. So that's it's a muscle, okay, that contracts. Uh, they have coiled shells, and that contains their visceral mass. It was mentioned as one of the 3 parts earlier. So we've got the foot, we've got the mass, visceral mass here. Okay. Some have vessels under their mantle that allow a kind of a what can serve as a lung. That's why it's in parentheses. It's not really a true lung for life on land. So that's why snails have to stay moist, but you can find them out in the soil. You can find them around in places as long as it's damp and wet, and usually close to some type of water supply. Because they have to keep moist. Class Cephalopoda includes octopi and squid. And here we see a foot that's evolved into tentacles. And we don't have much of a shell. The shell is reduced or completely absent if you have ever had the opportunity to um, dissect an octopus or a squid. Trying to remember if it's present in one and not in the other because it's been a long time since I've dissected those. But anyway, one of them or both of them maybe have what's called a pen. It's PEN. Okay, and that is a uh, rather flexible piece of well, it's almost like a fingernail. Okay, um, it's shell, but it's internal. Okay, it's inside the organism, and scientists look at that and think it's a vestigial shell. It's the remains of what was the external shell, and is just no longer serving its function as a protective outside covering. It may be doing something on the inside. Okay, just because it's like a quote vestigial may not mean that it's totally useless. We just don't understand the function of it currently. May have something to do with helping maintain shape, internal structure, that sort of thing. Don't know, but it's very small, very reduced. It's not what we think of as a standard shell. Okay, for sure, and it's internal, not external. Okay. Um, they also have powerful beaks and radula. Radula is this toothy tongue that uses to tear apart its prey. And you have to be kind of careful when you're dissecting these things because when you get into that area, if you're not careful, you can slice yourself on that. Okay. The beak is pretty sharp and the little radula is sharp too. uh, and they will puncture through a latex glove. I mean, if you really push on it and move against it, and sometimes in turning your dissecting specimen around and all that, if you're trying to really pry in there and see things, you can do that easily. Okay, because I have. So it surprised me. I didn't. I thought, oh yeah, that's yeah, but it's so small. Wouldn't be that sharp, right? Because the ones we were dissecting were not huge; they were small. But it's sharp. Okay. Class bivalvia includes clams, oysters, and scallops. So we eat these. These are one of the classes that well, people eat squid too. Okay. And octopi. If you've calamari. Okay. If you've ever had calamari, that's a octopus or squid, depending on. Sometimes I think it's a mix. Okay. Uh, and so you'd be, oh, gross, that's awful. But if you've never had it, you can't say it. Okay. One of my mottos when I was younger was I would never say that I wouldn't eat something or it was gross until I tried it. As long as other people had been eating it for, centuries and hadn't died from it. If there was no serious health risk in trying it, basically, I would try it. No matter how awful it sounded, I would try it. Uh, I can't say that anymore because I now have, and I probably was then. I just didn't know because it's genetic. And um, anyway, I'm celiac, and so I can't do anything that contains gluten anymore with the wheat, the rye, or the barley gluten. But um, prior prior to knowing that, I was like, yeah, I'll eat anything once. Okay, as long as there's no health risk, and see what it's like before I make a judgment on it. So I've tried calamari, and I actually sometimes like it. It's one of those foods where you gotta be in mood for it, because I've eaten it multiple times, and there's been times when it's like, "Oh, it was really good," and other times when it's like, "That was not, pleasing at all." And I don't know that they were cooked or presented any differently. I think it was just where I was at for what kind of taste I wanted on that day or didn't want. Okay. So, yeah, we eat more than just the bivalves. Okay. People eat conchs too, conchas, conchs in the uh, Caribbean islands. I understand this is a common dish, um, and maybe not as common. I don't know. It may be very common. I have no idea what the reference or the frequency of eating it is. But people who visited the Caribbean islands have told me that they've eaten it. I've been to Puerto Rico, and we didn't eat conchs there, but maybe I just didn't go to the right place or stay there longer. That may not be part of the Puerto Rican cuisine as much as it is more like um, Jamaica, Nassau, places like that. Those are the areas where I've talked to people who are from those areas or who visited, and they talk about eating the conchs. So it, may have some cultural things. Snails, of course, or escargot, um, and that's that. So we eat these all different these classes, but more commonly in your seafood restaurants here, you're going to find clams, oysters, and scallops. Okay, have the two shelves. So bi means two, and valve shell. Valve part. I guess. So you're opening. If you think about the two halves that open up on these, can help you kind of remember the name of that group. The shell is composed of protein, calcium carbonate, and mother of pearl. CaCO3 is calcium carbonate. That's um, limestone, chalk. Depending on what kind of pressure it has been subjected to, it can be really soft like chalk. It can be a little bit harder like limestone. And I think under enough pressure for a long enough time, it's what forms marble. Marble is calcium carbonate. Okay, so uh, mother of pearl is secreted by the mantle. And yes, pearls are formed from that. So the pearls formed when an irritating particle is placed between the mantle and the mother of pearl. So we always talk about the sand and the oyster, and it covers it up and makes this pearl. And yeah, it does that because it's an irritant, and it's just almost exactly like, well, it's at least analogous to us having an allergy. We get dust in our nose and we sneeze and start producing mucus to wash it out, and sneeze to blow it out, and that sort of thing. And what happens is that if the particle is not removed, we continue to secrete mucus. Okay. The oyster does the same thing. If the sand particle is not washed out by this producing of mother of pearl, which is a sticky, thick mucus type substance and isn't really good for washing something out. It does more of a coating effect. Okay. The mucus we produce doesn't, well, when it drips and runs, that does serve to wash a little bit, but a lot of that's more a coating. It's an extra layer of coating on our uh, respiratory lining to try to soothe, okay, cover up and soothe uh, the irritation. So, same thing's going on here. Um, it creates a smooth layer to smooth out the roughness of the particle, sand particle use for example. Uh, but there's still a bump there that's not supposed to be there. So we secrete the oyster secretes more mucus to cover that, wash it out, cover it, yeah, get rid of it, and it just gets bigger and bigger and bigger with the different layers of mother of pearl that get secreted on top of it. And at this point, you're probably, if you're following me along, you're going, wait, she's just telling me that pearls are nothing but oyster mucus, oyster snot. And well, the composition of oyster mucus or snot is much different from our composition of mucus, but yeah, basically, that's right. But it's really pretty. And so anyway, yes, that's how pearl is formed. And I like to try to draw that analogy to you, though, even if it is kind of gross on the human mucus aspect, uh, because it also helps you learn a little bit about uh, mucus in the respiratory system and our an allergic response. Okay, uh, so to dust and particles that you inhale, that's kind of the response to try to clear it out or at least stop the irritation from it. Okay, so similar process, different results, but uh, kind of similar goals, I guess. Maybe the process isn't even as similar, but the goals are similar. Okay, so between a class bivalve and a human. Okay, these are filter feeders. They attach or carried along by currents or scoot along uh, bottom areas of the uh, body of water that they're in, ocean, lake. Well, there's freshwater versions of these. Okay, freshwater mussels. The mussels in the creeks around here that you find, those are freshwater uh, bivalves. Okay, all right. So they can be in freshwater. They don't have to be in oceans. We usually think of the ones that I named as examples as ocean dwelling, but there are freshwater cousins. Okay, so the food and the water are going to enter through the in-current siphon. Mucus secretions help adhere food particles that's in that water to the gills as the water passes over the gills and mouth, and that traps the food there. So then it can be used. And then of course the anus, uh, where waste products are. Processed through we're going to empty into the excurrent siphon. The excurrent siphon will send all that out. We find two excretory kidneys that lie below the heart and remove waste from the pericardial cavity. Pericardial means cardia is heart, and so peri is like kind of sort of heart cavity. All right, and so we see also separate sexes. You have male mollusks or yeah, whatever. All three classes. Well, no, this is bivalves. This is just the bivalves. Okay, we're not back to class stuff yet. Okay, so you have uh, yeah, male oysters, female oysters, male clams and scallops and all that, and females. Okay, so they're separate. All right, phylum Annelida. Oh yeah, okay, that was all three classes on that. All right, phylum Annelida. These are our segmented worms. Annelida, Annel, I believe, is Annella. Maybe is ring, means ring. So these segments are a series of rings, looking things, uh, and that's most easily seen with the earthworm, which is an example of this phylum. So we have segmentation, and we have partitions that are called septa that divide the body internally. Um, we have a segmented coelom, that acts as a hydrostatic skeleton because the coelom itself is segmented. You can put water in one section and pull it out of the other, and that can help the creature move. Okay. So, earthworm, if you've ever dissected an earthworm, the place of interest in dissection is always this top part above the clitellum. Okay. Uh, and you sometimes will open the rest of it up, but this is the part where all the stuff of interest typically is. The instructors always want you to see that's got these little hearts, which I learned as aortal arches is what they were called. Uh, not given the distinction of being full hearts. They're little arch structures that act heart-like. Okay. Um, a little brain here. We've got a mouth. We've got a pharynx even. Um, the seminal vesicle. Uh, and the dorsal blood vessel and the ventral nerve cord. So, okay, dorsal means back and ventral means stomach. So, what we have here is we have the nerve cord actually running along the stomach instead of along in the back like we do in chordates. Okay. Um, so, and here we have the nerve cord is not a backbone. Okay. This is just the nerve. Okay. Running along there. But this is where we begin to see that structure occurring. Nephridium are like nerve type structures, kind of some sensory organs there. You get the coelom as the lining there around this body cavity. Oh, let's see. Yeah. Okay. And the crop is where food is going to be collected and processed. The ventral blood vessel also runs along here beside the ventral nerve cord. Okay, so those are all things that if you were to dissect this, you would be asked to identify uh, these items as part of your uh, lab. Okay, and then of course we've got the anus at the whole end of the body. Okay, kind of logically where we'd expect it to curve, the mouth, the anus, opposite ends. Okay. In this category, in addition to earthworms, we have marine worms, leeches, and yeah, I think a few other items we'll talk about as we go. So back into classes. So we have phylum and Elida. I'm going to break it down into classes again. I do that a lot here throughout the animal kingdom. We have class Polychaeta, and that means mini, mini bristles. And these are bristle worms. They have what's called parapodia. These are fleshy lobes that form appendages that can be used for swimming or crawling and also help function in gas exchange. So back here to this earthworm, anywhere we talked about primitive heart type structure. We've got brain, we've got a crop, which is your primitive stomach, if you want to look at it that way. We have kind of a nerve system, a circulatory system. Okay, and as the ventral nerve cord is you get these nephridium coming off of that. So where's the lung? Okay, we've got circulatory, digestive, um, sensory, nerve. I'm not seeing any type of respiratory organs here at all. And that's because these organisms still rely upon diffusion, okay, of gases through their body structure, their skin, okay, through their outer lining. Uh, and so that's why earthworms have to stay moist because as long as this stays damp, okay, stays wet, then they can exchange gases through uh, their, we'll call it a skin, okay? So that is how these function. And of course, the ones that are in the water, then they don't have any problems staying damp. But ones that live on land have to stay into moist soil. They have to stay near water where they can, uh, re-moisten. Okay. Uh, so if they get a little dry. The setae, which is what you're going to use to crawl, push themselves along, okay, are actually pretty exaggerated into bristles, and that helps anchor the worm as it moves. And I don't ask you questions about larval types and all that. We just don't have time to get into all of it. Okay, so earthworms. Here we have class Oligochaeta. Oligochaeta means very small. Setae translates as that. And so here we don't have a very well-developed head or parapodia. The setae are small. Okay. If you've ever dissected an earthworm, and one of the first things you are asked to do is to take your finger and run it down the length of the worm and find where you've got these little sets of bumps. Okay, those are the setae, and that is again what it uses to grip the soil. Okay, and work with that hydraulic segmentation of moving water into the segments and causing them to bulge. Okay, and then withdraw, uh, and so that helps. Uh, them move. Okay, so wiggle through the soil as you watch. It may not look like they're using any, feet type structures, but that's what these little setae do. Okay, they help grip. Uh, and so uh, that's the bottom. That's the ventral side of the worm, and so uh, that helps you to. Why you have to do that first thing is because that's going to tell you how to place the worm on the dissection tray. Okay, which side's facing up. Okay. So again, these earthworms, they are terrestrial as opposed to the marine ones, but they have to be kept moist, as I was saying, for the gas exchange. Oh, we have two earthworms mating here, and yes, they are hermaphroditic. But this again kind of shows you the reason that worms typically can't mate with themselves. They're going to mate with something else because the sexual organs are located in such a way that it's not feasible really for them to mate with themselves. And also, as I've explained several times in the genetics aspect, it's just not a good idea to reproduce asexually all the time. If you can, especially if you can reproduce sexually, you get more mixing of the allele possibilities and more variation in the offspring, and variation enhances chances of survival. So um, the question sometimes students will ask then is, well, what's the point of having both male and female sexual organs then if you don't have the ability to mate with yourself? And the point is that worms, think about where worms are on the food chain. What eats worms? Lots of things eat worms. Okay. Um, when you are lower down on the food chain and lots of things are predating you, eating you, you have to reproduce lots and often, or you are going to be eaten into extinction. And if your species goes extinct, then the food chain above you is going to collapse too. So it's important that um, animals that are down lower on the food chain usually have a short reproductive cycle, and the fact that they can usually reproduce very early. They don't have to mature long before they can reproduce. And then when they reproduce frequently and lots. Okay. Either they lay tons of eggs or they just lay eggs all the time type of thing. Okay. Uh, so with the earthworm, what this and any other hermaphroditic being, um, the deal is that anytime they meet up with another worm, they can reproduce, both of them. So the entire population is able to reproduce, not half. So where you've got separation of sexes in any species, it's roughly 50-50. You've got 50% male, 50% female, and that means only half of that population is able to reproduce ever. Okay. Here, 100% of the population has potential to reproduce offspring. And anytime they meet up, they can, and both go away pregnant. They don't have to wander along and go, oh, gee, you're another of the same sex. We can't reproduce. Okay, oh, find another worm. Yeah, oh, no, no, I gotta find another worm. It's doesn't matter. Okay, they both reproduce. They both go away pregnant. So increases reproduction rate, which is great because that keeps the food chain supplied. Leeches. This is class Hiradinia. And these are mostly freshwater. You can find some terrestrial and some marine. I'm not familiar with any, but the freshwater ones, I've had a few of these attached to me as I was a kid playing in creeks and things. Basically, stay out of still water. Still, and I don't mean the town, still into deep water in the shade. Okay, shady, deep, shady spots under willow trees. Yeah, that's they like to be there and they like to attach to you there. At least that's my experience. Um, and I don't know why it wasn't very scary. At first, I think I just thought it was mud on me, and I you know wiped it off, and it didn't want to come, so I wiped it off a little harder, and it came off. And then I had this little blood trickle down. It's like, well, what was that? And one of my older cousins was with me, and it's like, oh, it's a leech. And it's like, oh, okay. And what I should have done probably was go and treat that little bite with some alcohol or something and stay out of the water. But I was a kid. We were all kids. We just pulled the leeches off and stayed playing in the water. Just when I the second time I got some on me, it was kind of like, well, probably don't want to go back into that area of the water. Stay out of there. And then, um, somewhere later, either I read or told, or I went back and played and realized that it's in the deep still water under in the shade. And I think I've had that uh, coincidence as supported with reading I've read later. But yeah, they're interesting. Yeah, so you probably don't want to do what I did because that was a recipe for getting infection. I didn't, um, but uh, they do create a little wound there where they bite you. They are similar to other annelids, by the way, but they lack setae, the little rough feet-like things. The head is basically suckers. Okay, I have two modified suckers, one small oral sucker and a large posterior sucker. Um, they will attach to open wounds, but some cut through tissue. Uh, Ones that attached to me cut through tissue because, as far as I know, I didn't have any open wounds. Unless maybe I had some like chigger bites that I don't recall having, but I don't think I did. um, Not up on my legs. Okay, this was up on my legs where I found them. My chigger bites were always right around my ankles, and I don't remember any attaching there. Uh, so what they do though, and why a wound of theirs is likely to be infected if you don't treat it and you go back into dirty water, is because they produce what's called hirudin. It's a powerful anticoagulant that prevents clotting during feeding. Uh, so typically, I'm a person whose blood clots pretty quick. When I have to prick my finger for doing the blood examples, I think I mentioned that in class, talking about hypotonic, hypertonic, and isotonic solutions, and dropping the blood, pricking your finger and dropping blood onto a slide. Um, if I had to go back and get a second drop of blood, I had to prick my finger again, always. Whereas some of my classmates didn't. They just squeeze again and out comes some more. And it's like, wow, mine's already closed up. I can't even tell where it was. Uh, and so just, fast clotting. Uh, so under normal circumstances, I pull something like that off, it's going to clot really fast and I be in the water, no problem. But here, they've been injected basically as anticoagulant into us to keep our blood flowing. So even if you're fast clotting blood like I am, this probably keeps that from happening as fast and therefore increases the chances of that um, wound site getting stuff in it from dirty water. Okay. Uh, and then if your blood doesn't clot all that fast, then it's going to even prolong the opportunity for you to get that. So if you or children of yours or whatever play in the water and get leeches on them and all that, you probably need to not let them get back in. Okay. I managed to be fine. It was okay, but it probably wasn't a good idea in retrospect, now that I know more than I did then. Hirudin is used in medicine, though, for that purpose, to keep, not for infection, but to keep the blood flowing. Uh, and it's used during operations where they don't want blood at this wound site to clot up. Okay. Um, and leeches, there are such things as medical leeches, and I've only read a little bit about them. I find the idea intriguing. From my understanding of them, they they raise them laboratory. They're very clean, sterile environment leeches. They're not going to bring disease into you, obviously. Uh, but they will use them uh, during surgeries and place them around the incision sites to suck up the blood. And you may go, that sounds disgusting and gross. And oh no, I hope nobody ever does that to me. Well, I've seen some pictures in articles that I've read about this. And I'm thinking that if I were asked and given the option over surgical sponges or having leeches used during operation, I think I would choose the leeches. And here's why. The pictures where they've got the leeches there, the incision site is very clean. Okay. There's not a lot of blood and bloody sponges and gauze pads and stuff there. Okay. And everything that the doctor is working on, the surgeon is working on, is incredibly visible. Okay. And I've never operated on anybody, but I've done a number of dissections. And the hardest thing about dissections is having everything visible. Okay. And in dissections, the blood is gone. Okay. It's already removed. And even with that gone, it's still sometimes difficult to see to get to what you're trying to look at because you've got other tissues in the way. Okay. So imagine that in a surgery situation where you're trying to get to certain things, you got tissues in the way and you got blood all over the place too. And what they typically do is they, you know, wipe it up with these sponges and gauzes and things like that. And they stop there and they get full. They throw throw it over in the biohazard and put a clean one down. Uh, and that just makes it all messy. And every now and then you hear about somebody getting a horrible infection after an operation because they've had a piece of sponge or gauze sewn up inside them after the surgery, after the operation. And you're like, well, how did that happen? How could a doctor be so sloppy as to allow that to happen? A blood soaked gauze does not look that much different from bloody tissue in the body. Okay, looking at the pictures and watching videos of things and all of this, um, and it's a personal experience of, of treating wounds and stuff. You start looking at that, and it's like a blood soaked gauze does not look that much different from tissue that's immersed or totally bathed in blood. I think it would looking at those pictures. I think it would be very easy to miss um, a totally blood soaked gauze or sponge. Okay, so that's why I'm thinking, man, in contrast, the leeches to me look like better option, okay? Because it gives the surgeon a much cleaner place to work and uh, less likely leave something inside you that doesn't belong because you just can't see what they're doing for all the blood, okay? Um, so there, take that for what it's worth for you, but as long as it's sterile and they're not bringing any type of disease, why not? Okay, they're doing their natural thing. Uh, you want to keep the blood flowing because you don't want clotting to occur. And so they've got the natural Hirudin. Now um, we make a Hirudin synthetic version, okay, that's given as an anticoagulant as well, but it's based off the natural one that we find in them. Okay. Arthropods. These are a huge category, and this will be, I think, the last one. This presentation is getting a little long here, but Arthropoda is a group that contains over 6 million species. It's more than any other group, and more than a lot of other groups combined, even. I think Insecta itself is the largest one. It contains more than any other one combined, and all the others combined in this group. But and that keeps going because we keep finding more insects. So here's some examples of some of the arthropoda. We won't talk about all of them, but we have millipedes, tarantulas, paper wasps, stone centipedes, the Dungeness crab, lots of things. The characteristic of this group is jointed appendages. So we know from previously, pod means foot. Okay. Oh, I didn't talk about that with cephalopods. I just threw that name out there. Um, Actually, I didn't talk about the naming meanings on all of those. I should do that. Okay. Gastro means intestine, so gastropod, stomach footed. That's your snail. Okay, stomach footed. The foot's on the stomach, moves on its stomach, right? You see that. Cephalopod, cephalus means head. We've had that before. Pod means foot, so head footed. Okay, cephalopods are your octopi and squid, so your tentacles coming right off the head. You got the head, and it doesn't appear to have hardly any body, and then all of a sudden you got tentacles. Yeah. Um, so head-footed. Arthropod, joint. Arthro is joint. Arthritis is the inflammation of a joint, right? Okay. So this is joint-footed. Joint, joint feet. Okay. These organisms all have an exoskeleton. Skeleton on the outside of the body, composed primarily of chitin. Okay, we've seen chitin before. That was in our fungal cells. The fungal cell wall was made up more of chitin instead of cellulose. Um, and this serves to protect, anchor the muscles, anchor to the exoskeleton, and also the exoskeleton will prevent drying out of these organisms if they're not in the water. Because it's hard and unexpandable, though, it has to be shed or molted as the animal grows. So I think you've probably studied that uh, before with insects in high school, junior high, high school, somewhere life sciences or biology. You had to talk about molting of an exoskeleton. Uh, so we have three segments, three body part segments here that we're looking at. We have a head, a thorax, and an abdomen. And sometimes these are other segments are joined, are fused into regions, and these are the regions. So we have these three regional segments. And in some cases, the head and the thorax are even fused together in what's called a cephalothorax. So ancient trilobites, which you saw Sagan talking about in that very first film back at the very beginning of the semester, had appendages on each segment. So they're the earliest fossil remains of this phylum. Modern arthropods have fused segments for walking, swimming, eating, etc. And more examples, insects, arachnids, centipedes, millipedes, and they are all adapted for terrestrial life. Okay, so we have arthropods that are marine, the trilobites were marine, but modern day ones include uh, land species, okay, terrestrial species. We have a nervous system with a brain, a solid ventral nerve cord, compound eyes, and simple eyes, two types of eyes. Okay. So our first class in phylum Arthropoda is Crustacea. We have a calcified exoskeleton. Instead of just chitin, we're looking at some calcium deposited in it. And in that, we're going to see lobster, shrimp, and barnacles as examples. Oh, crab. Sorry. Didn't push my final return there. So here we have one pair of compound eyes, two pair of antennae, and three pairs of mouth parts. In crayfish or crawdads, depending again on your region, what you call that, the head and thorax are fused to the cephalothorax, as I mentioned earlier, and covered by a carapace. Hey, there's that word again. That's that word. I use that word throughout the semester. And the carapace covers the gills and protects the gills. So in past times, I've had students do dissections in class. Haven't done that in a number of years, but the dissections were a crawdad, crayfish, and a grasshopper. Okay. Uh, and I had the students find the parts, identify all the parts and external parts, and then students find the internal parts for each and make comparisons because they're both in the same phylum, but different classes. Okay, because this is Crustacea, whereas the grasshopper is going to be Insecta. But there are a lot of similarities. There really really are a lot of similarities. So you can just see how the adaptations are similar but slightly different for marine as opposed to land. Okay. We won't. I won't ask you any of those questions, obviously, um, because we're not doing that. But I do have the pictures up there so you can look at them. You can see for yourself that there's some similarities, quite a few similarities, and that it's the similar structures adapted for slightly different locations. So class Insecta has over a million described, totals more than all other species combined. Huge variety. Here's some examples. Okay, lots. I could keep, etc. Keeps on going. You could probably name 5 or 6 more if you think about it, of insects that you know. Okay. Um, Roly-polies or not, though. Ladybugs, yes. Roly-polies, no. Roly-polies belong in Crustacea. They're land crustaceans. Okay. So pill bugs. If you so what's a roly-poly? Roly-poly pill bugs, again, different colloquial names. So around 530 million years ago, there's evidence that centipede-like animals began to explore the world above water. This ties into an article that I have listed under this heading on D2L for you to look at. It's uh, Life Leaves Water for Land, something, I don't remember now the exact title of it, but that this is highlighted because at least, well, at one point it was a link, still is in mine, but when I put it up for you, it probably doesn't. I bet that link doesn't carry over, but it's a link to um, that article. Okay, but I took that article uh, and converted it to a Word document and put it into your uh, D2L so you can read it in case this link ever goes dead. Okay, so do that. Okay, read that. Oh, Evolution Out of the Sea. I'm pretty sure that's the article title, actually. Evolution Out of the Sea. So here's where this ties into our kingdom discussion, because a lot of what we focused on, obviously not insects, but a lot of what we focused on so far is marine based. We've got either lives totally in the water all of its life, or still, even if it's not in the water, the land based species still have to be close to water. They have to stay moist or damp. Okay. Insects are different. Okay. And this article here kind of talks about how some of those adaptations showed up in the fossil record, and how we can see that the dependence on water and moisture uh, uh, changed. Okay, how the evolution away from that dependence happened in certain structures forming. Okay, so insects. That's kind of where we, like I said, that's where the break seems to first show. We have 3 body regions again: head, thorax, and abdomen. We have all these sensory organs that you can see listed there. The abdomen contains most of your internal organs. Exoskeleton is lighter. It has less chitin than other arthropods, and you don't see calcium carbonate deposits in it either. Okay, so this is so they can fly. Now, not all insects fly, but a lot of them do. And the ones that do, they have to have a light body in order to be able to do that. So uh, reduced amount of chitin reduces the weight. So grasshopper is the main example of this. This shows, here's the body parts and stuff that I asked students to locate in a dissection. And these were the 3 main features that I always ask them to compare and contrast: gills for water, extracting oxygen out of water, and trachea for extracting oxygen out of air. Um, Ammonia versus uric acid excretions, and respiratory pigments versus simple diffusion. So we won't go into those details, but those are, again, because you didn't do those dissections. But I like to keep this slide in here because it gives you an idea, even if we don't discuss it in detail, it gives you an idea of, again, similar but different structures. Okay. I think gills and trachea are probably the easiest pairing here to compare and contrast without a lot of detail knowledge. Okay. We see this is adapted for water extraction of oxygen out of water, and this is adapted for extraction of one gas out of a mix of gases. Okay, so uh, yeah, the different locomotor systems and nervous systems, but uh, yeah, not gonna spend any more time on that. Okay, phylum Echinodermata. Ooh, I thought insecta was our last grouping. Okay, it is. It is because echinoderms and chordatas are deuterostomes. Okay, so I wanted to just do one presentation of our protostomes. Right. Okay, our mollusca, annelida, and arthropoda. Okay, and then come back and do another presentation about deuterostomes. So this is where we'll end it here then with insects, and you can go take a break. Okay, and I'll see you back for some echinoderms. There we go.